So hello everyone, I am Hridey Wason and I am here to present on topic gene expression and transfection. So what is gene expression? Gene expression is a process by which a cell can amplify or reduce its own functionality. Now you all must be wondering how does that happen. For that, let's look at the process of that. The process contains of two steps. The first step is transcription, which happens in the nucleus of the cell. And in this, the double-stranded DNA is converted to the single-stranded template of RNA, which is then transferred from the nucleus to the cytoplasm of the cell, where second process, translation, happens. In the translation, the RNA is converted into the proteins, which determine how a cell will function. Let's move on to the gene transfection. Gene transfection is a process by which we can artificially introduce a foreign DNA into a cell. Why do we do that? We do that to create a desired trait of the cells, and that is done by the transfection, and that is of two types, stable and transient. The transient transfection is a transfection which is temporary based. The cell does not accept the foreign DNA and it does not replicate it with own DNA. And the another one is stable. The stable transfection accepts the cell accepts the foreign DNA and replicates its own with the cell genome. What are the methods of transfection? There are three types: chemical transfection, biological transfection, and physical transfection. For the in the physical transfection, we use methods such as electroporation, magnetofusion, sonoporation, and gene microinjection to puncture the plasma membrane of a cell and inject the foreign DNA into the cell. In the chemical transfection, we use chemicals to help the foreign DNA to move into the cell easily. And the biological transfection, also known as viral transfection and transduction, uses a vector which is most likely a virus. So this virus helps the cell to diffuse the foreign DNA into the cell. What are the applications? So as the gene transfection becoming more popular technique, and we are accepting it to use in various medicine research, like biomedical research, stem cell differentiation, gene expression studies, and large-scale protein production. Now you all must be wondering, is there any link between them? Yes, there is a link, and that is when we introduce a foreign DNA into a cell. Now that cell accepts the foreign DNA and replicates it. So this is where gene expression kicks in. This gene expression will determine how this foreign DNA will produce proteins and help that cell to function like we desire. Thank you everyone. That will be all for the presentation.